So welcome. Thank you for listening on this new way of doing things. I do not have any feedback, so we'll have to see how this goes. So I will talk to you about the seasonal cycle of physical, biogeochemical, and biological properties in the marginal ice zone and farm strait. And I will try to point out some differences in the sea ice conditions during the growth phase and how that leads to different carbon production and export patterns. I'll also try to motivate this study a little bit broader, and I'd like to advertise a second talk. I was supposed to hold that at the Bedford Institute of Oceanography next week. Obviously, that has been cancelled. I presume if this goes well, we could recommend to hold it in a similar technological fashion on the originally scheduled date next week. So my name is Wilken-Jon von Appen. I'm a physical oceanographer. I've been at the Alfred Wegener Institute in Germany for 7 years now. So this study that I will talk to you about contains physical oceanography, but it's only a part. And there's a large number of collaborators who have helped me understand and make sense of the data set, but I am not an expert in any of these other aspects, biogeochemistry, biological properties. So if there's something that I'm saying which doesn't make sense, I apologize, and I would ask you for feedback to see where I can improve on this. Yeah, and maybe as a background, so I'm an OFI fellow here right now, and apparently it seems like I might stay in Halifax for a while longer than originally planned. This talk is also, as far as I understand, recorded on Twitch on this website that you're looking at. So if you have to run out or you would like to show this to someone else later on, you can watch it later on. Also, depending on how this questioning goes, we can try to do it at the end of the talk or in between. But I'm also very happy to interact with people via email or Skype afterwards. I think since we can't just do face-to-face in person, we have to use all these technological measures. So please feel free to contact me afterwards. Right, and to give you some context, the data that I will show you comes from a large grant that was given to our institute. It's maybe not the same size as the OFI, but it's a similar order of magnitude that we received in 2015, which was then spent on purchasing a lot of equipment that was available at the time and to deploy it in the Arctic Ocean, and to also employ scientists, engineers, and technicians to learn and to learn these measurements and to work on them. All right, so I will go to the next slide. As an introduction, what I will talk to you is about the seasonal cycle, and specifically going from the upper ocean, from the atmosphere, all the way through material exported that ends up at the sea floor, really trying to make the whole connection from the surface to the bottom. At low latitudes, that is somewhat easier to access, so we have some understanding of what these things are. Of course, in the open ocean, they're also somewhat limited. But in the high latitudes, you can get that from shipboard measurements, so we really rely on new sensors and samplers. Also, there's the big question how sea ice impacts the seasonal cycle. There's two obvious aspects in which it can do that. It could increase the stratification through melting of ice, and it can also impact the seasonal cycle by modulating the light penetration into the water column, either limiting it or then also allowing light penetration when ice melts or when melt ponds form. Of course, we have polar night and polar day, meaning that the light availability is completely shut off in winter. The other thing we know about the Arctic Ocean is that open water conditions may become more prevalent. The area that is affected by sea ice will decrease as part of climate change, and often that is called Atlantification. So a situation that is typical of the North Atlantic may be present in larger areas of the Atlantic Ocean than was previously the case. So my work is mostly focused on Fram Strait. That's the strait between Svalbard in the east. You should be able to see my cursor now, which I'm moving. And on the western side, this is the northern tip of Greenland. So to the south of this area, we have the Nordic Seas, the Greenland Sea, and to the north is the Arctic Ocean proper. This is the only deep connection between the Nordic Seas and the Arctic Ocean. Of course, the actual connection at depth is more limited by the Greenland-Scotland Ridge. Here I'm showing the isobath. So we have a shelf in the east and on the west, and then a deep ocean deeper than 2,000 meters in the center. This area is characterized by three main currents / current systems. It is the northward flowing warm and West Spitsbergen current, and on the east there is a southward flow of colder water and the so-called East Greenland current. Some of this water moves to the west, in what we call the recirculation. So water enters, makes its way to the north, but some of it makes the way to the west. That leads to a semi-stationary ice edge. So what is shown here in color is the number of days, or the percent of days that the ice concentration exceeds 20%. So in the eastern part here, where there's an inflow of warm water from the south, the time between April and June is entirely ice-free, whereas there's export of ice from the north, so this area is entirely ice-covered, and then there's a fairly narrow region in the middle, and it also doesn't change between the seasons, so the location where the ice edge is located doesn't move as far north or south as it does at other parts of the Arctic Ocean or the Antarctic. So I'd like to talk to you a little bit about what we know about this current, the West Spitsbergen current. This is data from a previously published study. We have had a mooring array along the center of this strait. On the right of the figure here, you can see the shelf dropping to relatively deep areas. And in August, this area is highly stratified. It can be seen here from the colors. This is from shipboard CTD stations. The dots are moored observations. Whereas in February, the stratification is fairly well mixed, not entirely unstratified, depends on the time. So this is actually the Argo float coverage in this area up until 2016, all the floats that were in this area in that month. We don't know very much about the winter there. So this area is characterized by Atlantic water. That is the water that's coming from the south. It's moving northward. It is high in temperature, salinity, density, and nutrients. And it is also characterized by water that's coming from the north, from the polar water, which is low in temperature, salinity, nutrients, and density. And what actually happens here is that this water partially moves from the west to the east in the spring and covers this area to increase the stratification. The majority of the stratification is not due to temperature, but to salinity. And that results in a seasonal cycle like this. The different colors are different moorings. They are, as you can see here, these dots. Solid lines are 50-meter depth measurements, and the dashed lines are 250 meters. So the water column, there's no difference in the density between these depths in winter, and then throughout the summer, stratification sets in as the density at the surface decreases. In this area, what also happens is that as these two water masses mix and intermingle, so-called filaments are generated, in this case submesoscale filaments, and I'm not going to go into detail here, this is more the advertisement. So I would like to show you on the right here an image from satellites in the center of Fram Strait. This is water depth more than 2,000 meters. And what you can see here is a scale bar of 20 kilometers on the bottom, and the image itself is radar reflectivity. Over here on the right side, it's open water. You can see internal waves. And then all the other structures, this is a big ice flow. The rest is ice. And ice is being moved around by the ocean currents and basically shows us the ocean dynamics below. So we did an experiment here, actually at one of the cruises that turned around the mooring data, which I will show you later, in this area that is marked by the magenta box. And what you can see here, there's a straight line from roughly here to roughly here that is 50 kilometers long, and very narrow, thin sea ice that has been moved along. So we try to explain and understand what that was and what the oceanic dynamics are that are associated with this, and how this affects the hydrography and the chemistry, biogeochemistry in that area. So I was supposed to, my plan was to talk about that at the Bedford Institute of Oceanography seminar next week, and to combine this together with another recently published study where we really use these towed observations with which we can now also make statements about biogeochemistry. We also proposed to incorporate this methodology and equipment into the OFI project, the Northwest Atlantic as a Climate Ocean, projecting future changes in productivity and the biological carbon pump. Part of the reason for me to visit as an OFI fellow was to discuss how to do this integration. Of course, it depends a little bit on if and what this project will be like. I think I would still like, I definitely think we will still do this discussion. And I think knowing about the methodology from these papers would be quite helpful. So either I'm going to present that to a group that's involved with this project, or you can tune in onto the seminar next week. So let's go back to the seasonal cycles. And this is from another study. This is the Laptev Sea. So previously I was talking about Fram Strait, which is on this side of the Arctic Ocean. In the Laptev Sea, this is a shelf sea, there were 2 years contrasted, 2018 in blue and 2015 in red, from the timing of different things. And one thing that becomes clear here is the sea ice cover is shown here essentially disappeared, in this case, at the end of May in 2014, and lasted for another month and a half until the mid of July 2015. This was then followed by the increase in measured chlorophyll or fluorescence after the disappearance of the sea ice. That also agrees with changes in stratification. Once the ice disappears, the ocean becomes stratified, allowing for production. In the Greenland Sea, so south of Fram Strait, there was a study 2 years ago based on Argo floats, biogeochemical Argo floats, that tried to contrast cases of production under ice and partial ice. So here sea ice concentration between 50 and 100%. So it's not 100%. It's there's also light that can penetrate the ice or the ocean, can penetrate into the ocean away from in between individual flows. And what was shown here is that there is production underneath ice in the area in the time when there's still ice present. And then afterwards, when the ice disappears, then open water subsurface chlorophyll maxima is produced. And that also points to the fact that, of course, a lot of these dynamics would be missed from remote sensing. So satellite remote sensing doesn't work very well in the presence of sea ice. But it also cannot pick up on subsurface chlorophyll maxima. So once there is open water, in principle satellites could see it, but they're not going to see the subsurface chlorophyll maxima very well. The study also, another study that came out recently contrasted in this area the years when ice was bigger, or when the ice export increased or decreased in this area. So in years with more ice export, there's actually also more ice present in this red area. I would like to point out that the measurements, which I will discuss in detail more, are actually just located to the north of this map. And then there's also years with less ice export from the Arctic, then that results in less ice in this area. So then there's a sort of a line of argument. When there's a high ice flux in farm strait, then there's high ice concentration in the Greenland Sea, and that results in stronger stratification due to meltwater and an early and larger bloom. So here's the progression of production, and the peak moves around by more than a month between different years. Here was also argued, so 2017 is a year with more than average ice flux through Farm Strait. 2018 was less. And there it's also argued that in the Greenland Sea anyways, this year appears to be different because it also has an early and large bloom, even though there's less sea ice exported. So they speculate that this might be due to temperature stratification rather than meltwater stratification. So now I will go back to Farm Strait. We had moorings here in two locations, one in 1,000 meters water depth and one in 2,500 meters water depth. And we had them in the water since summer 2016. So we have two spring realizations that I'm going to discuss here. That is 2017 spring and 2018 spring. We are continuing the data collection, so we in fact have another set of data sitting on our desks from 2018 to 2019, and instruments are in the water to be recovered in 2021. However, as you can see, there's a lot of information, and at some point one has to sit down and not wait for the collection of more data from the next year, but just use with what is available. So I'm just focusing on these 2 years to not essentially be overwhelmed. What I'm showing you here now is to characterize those 2 years. The green lines are the weekly averages of the 20% ice concentration, which we take as a proxy for the ice edge. And you can see that they are a little bit removed towards the north or west from the average, from the long year average, but that's not that far away. But in principle, in fact, it's also quite far away from both of these mooring locations. In 2017, that was different. The ice edge was actually much further south, and was essentially over one of these moorings, but it was still removed from the other one. So the way I'm trying to tell you the story now is to focus on the observations that we have at this western of the two, which we call HG4. And there we had in spring 2017, we had intermittent ice, whereas in 2018 we had open water. Whereas at the other location in both years we had open water. So we can contrast to make try to disentangle spatial from temporal differences, and essentially taking this F4 as a control. I will not show you this data, it's available and I'm happy to discuss about it. to see what are the differences just from interannual variability and what is the differences due to ice or not ice present in this region. Okay, so now we get to look at a number of time series. They last from July 2016 to September 2018. And what is shown here in the first panel is the depth at which we actually have measurements. So the blue is roughly 25 meters, the red is roughly 50 meters, and then we have in green data roughly in 250 meters. But due to the currents, these moorings actually did move vertically at times. So sometimes we are not in the euphotic zone anymore with the top measurement, whereas otherwise we are in the euphotic zone, but we are also not exactly at the surface. I should maybe mention two things about that. In terms of trying to get time series observations from ice-covered areas, in principle, people try to deploy these things such that they can recover their instrumentation. And to do that, you want to be in a safe distance from the moorings, from the ice, which is often deemed as 50 meters. So in the past, we have not had much information of shallower than 50 meters. Here we were brave enough to go with the top of a mooring to 20 meters. And we did recover our instrumentation, but we were not brave enough to go shallow. And in particular, you cannot have surface buoys because they will just be whipped off by the ice. So that limits the interpretability a little bit compared to this being proper surface measurements, and also this compared to being profiling measurements. But we can still make some inferences from this, I believe. And yeah, so the strength of this is that we really cover the different disciplines. So let's walk you through this. The yellow is here the distance to the ice edge. So I mentioned in 2018 the ice edge was not over the mooring, but it was never very far away. So it was on the order of 50 to 100 kilometers away. Whereas sometimes in spring 2017, ice was over the mooring, and then the ice concentration went up to these values shown in black. So it went up to 75% at times. It also went down to near zero in intermediate. So this is not the typical pack ice central Arctic condition. This is really marginal ice zone dynamics and situation, where sometimes you have ice, sometimes you don't have ice. Even if you have ice, it's not super densely packed ice. And also in this area, it's often small flows compared to the central Arctic. Now I'm going to show you temperature in the water and in the air. So the water are these colored lines. They are not showing very much of a signal except for the seasonal signal, which we sort of know from this area. It does become temperature stratified in summer a little bit, reaching up to temperatures of 6 degrees. That's not very warm, but for the Arctic, that's reasonably warm. And then in winter, it reaches not much colder than 2 degrees Celsius. So that area is not covered by real polar water, which is near the freezing temperature. Yeah. And then the yellow line here is actually the air temperature in this area, with a broken axis at -2. And what you can see, just to start with 2018, air temperature is above and below freezing every once in a while, and then from March onwards until the end of April, it is below freezing and surface freezing, and thereby extracts heat from the ocean, making it unstratified or continuing the destratification. In 2017, the situation is a little bit different in that there are also times in intermediate when there's no cooling, but the cooling does persist for longer. It actually persists until the end of June. So it doesn't allow for as much of restratification. Here I'm showing the concentration of polar water. So we're assuming there's either a warm, salty Atlantic water or from the south, or water that comes from the north, which is cold and has much lower salinity. In 2017, intermediate, for intermediate times, there is a lot of polar water, but then it goes away again and is essentially Atlantic water. And of course, the shallower one gets, blue here being in 25 meters, that's where polar water is present, whereas at depth, the Atlantic water is always present. That is not so much the case in 2018, where it's mostly Atlantic water up until the cooling stops. Then there's a little bit of lower salinity water, but not very much. So from temperature and salinity, of course, we can get density. We don't have profiling instrumentation here, so this is all we can do. We can look at the differences in density between the different depths. When the density that's high up in the water column is low, then it's stratified. When they are essentially the same value, as you can see in January, February, then it's unstratified. And what we can infer from that is that the mixed layer must have been at least as deep as our lowest measurements. And that is this black line. So in winter, we have deep mixing 250 meters, and then it gets shallower drastically and basically becomes stratified after early May. That's the black line here. The black line in 2017 goes from being intermittently very shallow, and what we can infer that there's at least, so we can only look at the difference between 50 and 20 and 25 meters, and if they are different, then we know the mixed layer has to have been shallower than 50 meters, so fairly shallow, but then it becomes deep again, back and forth, and that back and forth actually persists for much longer, for a month longer in the season than it does in 2018. And what is also can be seen here that in this area, the stratification that does set in 2018 is not thermal stratification. The temperature doesn't change very much between the different depths, whereas we do get water that has lower salinity at shallower depth. So different from this previous study that was in the Greenland Sea in 2018, that the stratification we see here is not meltwater stratified, but yes, meltwater stratified, but not thermally stratified. So now let's look at the non-physical parameters, or the parameters that are going beyond my personal understanding of physical oceanography, let's put it that way. We have measurements from light. This is photosynthetically available radiation. At 25 meters, every hour we have a measurement. These are the blue dots. In polar night, there is no light. There's light at the detection limit. There's a constant line here. And then in the transition period, we do have high values during daytime, and then still at the detection limit at nighttime, whereas then in polar day, it's always above the detection limit. If we average that to an average daily value, to remove the difference between day and night, then we get this green line. And I'll explain that in a minute, what we see there. But let's look at the other curves first. And that is the yellow line is the surface incoming solar radiation, which can be converted to photosynthetically available radiation. So that's really what arrives at the surface above sea and above the water and potentially sea ice. But what is relevant for the growth of phytoplankton is how much light the phytoplankton receives that is in the water column on average, and that's this red line. So we, using our crude estimate of the mixed layer depth, if we assume a constant extinction throughout the water column of the incoming light, then that gives us the average value that a phytoplankton cell living in the mixed layer would experience. And in 2018, it's really quite obvious that the light, even though at the surface it increases, because the mixed layer is deep, the light remains low, and then as the certification sets in on essentially the May 1st, then the available light increases by 1.5 orders of magnitude. So that's really then when the growth is not limited anymore. That's the red line. That is what is available. In relation to that, you can also see what is actually measured at 25 meters, and that drastically drops at the same time, which is essentially due to the fact that phytoplankton then grows and shades light from getting even to 25 meters. So you can see there's a quick drop here. In 2017, the increase is again more back and forth. Sometimes there are stratified periods with higher incoming light, and then it goes back down when it becomes unstratified. There's also an apparent decrease in the incoming light, in the light that is arriving at depth, but a little bit delayed in times. So we do have chlorophyll measurements also at 25 meters. However, the instrument failed here, so it only restarted. The numbering only restarted here. In 2018, you can see the low values in April and then high biomass as the stratification sets on. I have looked at this logarithmically, so there is some growth in April, but it's still the exponential growth rate increases on May 1st, so it's not a constant growth rate that we see here. The dots are satellite chlorophyll estimates, showing that there is also some chlorophyll in the surface ocean in the 2017, but again a little bit later in the season. AOU, apparent oxygen utilization, is we take here as a measurement of the biological production of oxygen, which is what takes place here as the bloom sets in. So at these two depths, blue and red in 25 and 50 meters, the values are at winter values. We're not quite sure why they're non-zero, but we have some assumptions why they should be non-zero, and also why they should be a little bit higher, even greater at depth at 250 meters. But as the bloom sets on, the phytoplankton releases oxygen, meaning AOU becomes negative, and we're getting this bloom progression seen in this biogeochemical parameter. The growth in 2017 was much less. You can again see that AOU goes down, but it goes down much slower. There's one intermittent event when it's low, but the trend line is much weaker. And that basically really shows that we had this light limited situation until May 2018, whereas the ice affected year was sort of slower, delayed, interrupted bloom that then sort of started in June and went on from there onwards. We have measurements of inorganic nutrients too, and here I should mention one technology which you guys not everyone might be familiar with, and this is a so-called remote access sampler. So it takes water samples of 500 milliliters. You can program when it takes them. We took them roughly every 2 weeks. So you get 48 water samples, which you can then analyze with laboratory technology that for which currently sensors don't exist. This was poisoned with mercury chloride. And then we also had a sensor for nitrate. So let's look at blue, which is nitrate. In the second year, this is from a sensor. We didn't have sensors in the first year. Winter values, which are fairly constant as the mixed layer is deep enough and the mixing goes down into the constant values at depth. As the bloom sets on, nitrate decreases rapidly over a course of a month and a half to get to near zero values, which is when then the bloom becomes nitrate limited. The phytoplankton, or the chlorophyll decreases, and also actually more light gets into the water again, so that's basically the end of this swing bloom, which then crashes and actually also releases some nutrients again, and respires, as you can see in the increasing AOU. The inorganic nutrients, in the first year, these are from bottle samples, so nitrate in blue, silicate in purple, and then we have phosphate and nitrite. Nitrate decreases a little bit throughout this bloom period, but it doesn't get nearly to limited values, unlike the open water case. But we do see nitrite increasing throughout this progression, and my understanding is that this is a byproduct of biological production, so it does indicate that there is some growth taking place. And of course, the system is not silicate or phosphate limited. We have partial pressure of CO2 and pH here in a reversed scale, so that the wiggles essentially align. We can also see the winter values fairly constant, and then a decrease in PCO2 as CO2 is taken up to make biomass. And then the respiration takes place after the main bloom. Again, not a clear signal in the first year, where there was some production, but it was much less. So let's look at the optical backscatter was something I wanted to mention. In red here we have measurements not of the average optical backscatter, but of individual peaks, maxima, that indicate when there's large particles in the water. So this is at 25 meters. When there's large particles in the water, they are probably aggregated, or one can hypothesize that they are aggregated big particles which have the ability to be exported. In this bloom case, that takes place about a month after the onset of production, close to when nitrate becomes limited, and also as the standing stock decreases. So it's possible that this is when the export starts. There's also export or suggestive aspects that are suggestive of an export taking place late in the season here, where we have measurements, and then also in the middle of winter. So let's look at the slopes here. So this is a scatter plot in 2018 in the open water bloom case, where we have this data of nitrate versus chlorophyll for the first month and a half from the main bloom, May to June. This is essentially from this color to this color, and the scatter plot follows the best fit, which is the black line, and then in the later part, mid-June until early August, it's the dashed line. So in the early part of the bloom, there's a very strong essentially linear correlation, an R-squared of 0.6, and the slope is minus 0.9. So colleagues have told me, just like sort of order of thumb, you can say that for 1 micromole of nitrate per liter, you can grow 1 microgram of chlorophyll per liter, which this suggests to be fairly appropriate, actually, based on this data. And of course, in the later part of the bloom, that doesn't hold anymore. We have the same here for apparent oxygen utilization and chlorophyll. A fairly good negative correlation here again. And then between AOU and nitrate, it's also a fairly strong correlation. And that in this case means that for each nitrate taken up from the water column, 6.8 oxygen molecules are released within the wet field ratio. Not necessarily unexpected, but still nice to see here, I believe, from independent observations, of course. So then let's look a little bit what the community is made up out of and how this then goes through the water column. So we have observations here again from water samples from the DNA, eukaryotic DNA and bacterial DNA, and we have left it here at a fairly high level of just breaking it down into diatoms, dinoflagella, and everything else, and then broken it down into one type of bacteria that dominates, and then another one, bacteroidetes. This is at the mooring I was showing you before. For the second year, we don't have the analysis finished yet, and this is the first year at the other mooring location, which is open water. And what you can see there is really a diatom bloom. So diatoms are relatively unabundant, and then as the biomass increases, they are taking up 60% of the biomass or of the DNA, which so what we see there is a diatom bloom, whereas in the ice-covered situation, there's no clear signal; it just wiggles around. In both cases, though, these bacteroidetes, which we believe to eat phytoplankton, increases as that is available for them to eat. We also have acoustic measurements from an ADCP, which is essentially telling us the proxy for the abundance of zooplankton. The contrast we see here is the zooplankton abundance seems to be dropping throughout the season until the phytoplankton bloom starts, and then they actually happily grazing there. And what that also suggests is that the onset of the standing stock we see in early May is not due to the reduction of a zooplankton grazing pressure. So basically, there's not grazing pressure in April that would have inhibited the increase in standing stock, but it's really the effect of stratification that explains this. And again, in the ice-covered year, there's no strong change from pre to post bloom, or pre-bloom to bloom to post-bloom. And we have export data. It's a little bit of a busy figure, so I'll try to walk you through this. What we have here in 200 meters in black and 1,200 meters in red is export as a function of time. The units are essentially uncalibrated. It's more qualitative than quantitative here. And then we also have measurements in blue from a sediment trap above the seafloor 2 meters above. That gets some of research suspension. But what we can find here is that we actually do have some export in April before the main bloom starts. We're not quite sure what that means. But then the majority of the export actually takes place in June, so about a month after the onset of the bloom, and it's a little bit like when you're shallow in the water column, it starts a little bit earlier, but the temporal resolution of these things, of course, is not as high as hourly. And there's also a late export event apparently, whereas in the ice-covered year, the export is much delayed. The maximum export actually takes way late in the season, but essentially before or around the onset of polar night. We also have measurements of oxygen consumption in the benthic, in the benthos, in the top several millimeters of the sediment. This is this green line. The up and down are not as obvious as you might believe, but there's an increase in June, about a month after the onset of the bloom at the surface. Now in 2,500 meters depth, we can see an increase in the benthic consumption, which is benthic respiration, which is due to the arrival of organic matter there. And if we just assume like the bloom started in the early May and then it made it within a month to 2,500 meters, that's about 80 meters per day as a vertical settling velocity, which is reasonable. We also have, in the second year, we have photos of the seafloor once a day. And what was quantified there is what that seafloor is covered by. So it can either be barren seafloor, or what we found that it was covered by green fluffy material, which is fairly big aggregates that are rich in chlorophyll. That is what this green curve is. So essentially until the end of May, it's barren seafloor. And then the seafloor is covered by this green fluffy material. We also have another type of export event that is white material. And we don't exactly know what it is, but it appears to be much smaller, much less aggregated, and appears to be associated with the export event that's seen in the late season here. So essentially the remainder of the surface biogeochemical community that relies on light gets exported at the time when polar night sets on, because there's no light anymore. We also counted the individuals that were seen on the photos. So we see benthic megafauna here as these, in the first graph here, shrimp, isopod, in the second graph, and fish. And what can really be seen, it takes a little bit of time after the arrival of this detritus material, but then benthic megafauna is visible and probably contributes to eating up this material. There's much less of a response for this white material from the benthic community. So I'm almost done here. So some questions I have, and if anyone has inputs or suggestions on that, I would really appreciate that. That's why I'm here, or was supposed to be in the office at Dell. So essentially what is not clear to me really is how much processing or data quality control is required to be able to publish a story like this, which is really about timing and qualitative aspects and not so much about the absolute quantitative numbers that are shown here. And then I think we also still need to work a little bit on narrowing down exactly what is the story and the audience that this is interesting to, and then what are leads for analysis and suggestions. And I realize I'm a little later than I expected, but it's very difficult not to get direct feedback from the audience, so I apologize for that. So with that, I'd like to summarize. We had these two different years. 2007, intermittent ice cover, delayed slowed bloom, onwards no complete nutrient drawdown. 2018 was open water, stratified, unstratified meant to stratified conditions. The bloom really was from May 1st onwards. The nutrients were consumed and were exported to the seafloor 1 month later. We had this F4 for ice-free comparison. We have ongoing measurements. And I was supposed to be at Dell until April 3rd. Looks like I'll be around Halifax for longer. And of course, I'll always be available for discussions on email and Skype. And I would like to, thank you for listening. I think the plan is that people who have questions are welcome to type them now, and I can try to address them. I would read out the question, think about it myself, address them, or we have to carry this discussion to email and Skype. And with that, I'll stop talking because currently I see something from David. I guess we also can see whether someone is typing a question. So I guess we'll wait like maybe a minute or two. And otherwise, if someone says they would like to type a question, just say like, I'm typing, and then you can type it at your leisure, and then we can wait for that. Aaron, a methods question for you. For the DNA analysis, you filtered the 500 milliliter sample that were remotely collected and HD killed once they were back in the lab. Right. Okay. So yeah, that's basically what happened. So the samples were collected over a period of a year. At the same time where these nitrate measurements or nutrient measurements were done. The bags were collected out of the instrument. Then they were filtered, and I would need to look up what the details of the filters are. The filtrate was used to do the. No, I believe there was a split done before for inorganic nutrients, maybe 50 milliliters. The rest was filtered, so about a little bit less than 500 milliliters. That was filtered, and then those filters were brought to the lab for standard 18S and 16S analyses. And what I really like about these RAS is that these remote access samplers is that they allow analyses that people like to do in their labs, which we will not in the foreseeable future be able to do on sensors. So in that sense, they are a nice tool. We also had so-called phytoplankton samplers deployed. So they are actually filtering in situ and just bringing back the filters, which are also poisoned. My colleagues have found that they can do the same analyses from the water samplers as they can do from the phytoplankton samplers. So that we're actually thinking of discontinuing to use the phytoplankton samplers because they're not giving us an added benefit. I mean, essentially, they allow to sample a larger volume or to filter a larger volume, but that so far has not been an issue. Yeah, so I understand that, I forget who, but someone in the OFI project has a phytoplankton sampler available, and I had mentioned this a while ago that I should put you in touch to, yeah, I'm going to talk about this later. You mentioned data quality control. Was there any data quality on the data that you presented, or is this purely raw data? Right, so it depends. For the physical data, temperature, salinity, that is data that we process the same way as we would do any other mooring data. So I would consider that quality controlled. For the data that you can see currently on the figure, we don't necessarily exactly know how to do that quality control, and for the nutrient samples, my colleague does have data control, so that's compared to CRM's reference standards. But for the other ones, it's actually very hard to get anything out of it except for the qualitative thing. So for example, also we don't know whether the offset here compared to 0 is real or not at the ARU. Yeah, another aspect might even be, rather than trying to answer whether that's publishable here or not, it's more one needs to take that one needs to essentially decide how important it is to be able to do this quality control afterwards for the story that one wants to tell before doing these deployments, because then you need to be able to do that preparation as you deploy these instruments. And a little bit logistically, we were overwhelmed had to buy all these, had to like understand how to program them and where to put them. And then the second step, how to program, how to get yourself set up for quality controlling them and also knowing exactly what you need to do. I think we're now in the process to be able to decide what needs to be done so that we could do that for the future, if that makes any sense. There's something from Dave Greenberg. Last slide, nitrate, middle plot. The more continuous right looks noisy. Are there error bars for the left part? Right, so the left part, the first year, that is from samples, so from water samples. The right one is from, and they have a resolution of roughly 2 weeks, a little bit higher in the spring than in winter. This is hourly data, which is why, from a sensor, which is why it's more noisy, because it actually also samples not necessarily noisy signal of the sensor, but vertical and horizontal spatial heterogeneity in the ocean. There's spatial gradients in the ocean which are advected past the sensor, which we probably don't know much about, but are being picked up here. So that's why they look different. With respect to error bars, they do exist for the samples. I haven't plotted them here, but they are small in the sense that you would see them if I had plotted them, but they wouldn't reach much beyond the range of these dots here. All right, well, thank you for listening. Thank you very much.